Hello, thanks for joining us today for this Ultivue webinar. My name is Renee Suleiman, and I'll be the moderator today. In this webinar, Yvette Cajigas and Dr. Angela Vasaturo will be delving into the validation of a multiplex immunofluorescence workflow that provides a simple approach to immune profiling and evaluation of precision therapies. Yvette will be leading this discussion, with Angela in the background for support with questions. Yvette Cajigas is a board-certified histotechnologist with nearly 2 decades of experience in anatomic pathology. Currently, she is Director of Product Development at Ultivue, where she leads a team of scientists in developing a robust services portfolio that meets the technical needs of Ultivue's customers. She has served as a technical lead at clinical, academic, and contract research pathology laboratories, and has extensive expertise in histologic assay development and multispectral digital pathology. Angela has over a decade of experience in translational immune oncology and is a leading expert in multiplex IHC techniques, digital pathology, and tissue imaging. Currently, she is Director of Scientific Affairs at Ultivue, where she guides customers through the assay development process, beginning with scientific consultation all the way through image analysis and data interpretation. She is also responsible for establishing valuable scientific partnerships and generating internal and external data that demonstrate the quality of Ultivue assays. Without further delay, I'll turn the time over to Yvette.
Thanks so much, Renee, for the introduction. Hello, everyone. My name is Yvette Cajigas, as Renee mentioned. I'm the Director of Product Development and Services here at Ultivue. Prior to joining Ultivue in 2020, I worked in academic medical centers and CROs supporting assay development and validation, and in support of clinical research and also in support of clinical diagnostic services. Since joining Ultivue, I've had the pleasure of leading both development efforts for our commercial offering as well as custom development work for our services lab. Most of us are here today because our work involves biomarker development and validation, or because our research is supported by the use of these biomarker signatures. The goal of this kind of work is, of course, to first discover a biomarker signature, validate that biomarker signature, then demonstrate its clinical utility via clinical trials, and ultimately to move that biomarker signature into the clinical diagnostic space where it can have a real impact on patient outcomes. The tricky part, of course, is that there's an inverse relationship between the number of biomarkers and the number of patients enrolled in a clinical study. That is to say that in order to be confident in a biomarker signature, you often require multiple biomarkers to define that signature, particularly in that early phase, that early discovery phase, as you're defining and refining your hypothesis. But then as you move further down the biomarker development pipeline, those cohort sizes get larger and larger, making it harder and harder to interpret the existing data. For the purposes of my talk today, I'll center my discussion on analytical validation, the second bubble here, delving into our methodology for validating and highlighting adaptable multiplex fluorescent workflow. And I aim to instill some optimism regarding the prospect of enhancing the feasibility of clinical validation and ultimately the clinical utility of multiplex assays in the near future. Before I delve into our approach for validation, I want to briefly summarize our technology and the specific platform that we have validated, the OmniVUE platform. InSituPlex technology, shown here, otherwise known as ISP, allows for the visualization of up to 12 protein targets of interest. All the targets are placed on the slide at once, at which point they bind to their target. This means that unlike other cyclic or TSA-based methods, our technology does not require antibody stripping, and that's particularly beneficial as it ensures that tissue stays intact, and that allows for same slide H&E further downstream. Once those antibodies have bound to their target, all the antibodies are amplified at once. And that amplification increases sensitivity over other proteomic approaches like mass spec or other non-amplified methods. Then the corresponding probes are added, which bind to the elongated barcodes. It's important to note that those fluorophores that are added are spectrally distinct, so that eliminates the need for spectral unmixing. This workflow can be carried out both manually and automated on the Leica bond. And what the OmniVUE platform allows is for researchers to design their own biomarker panel from a core library of clinically validated clones, as you can see here. And I will describe the way in which we validate those biomarkers next. So what does that validation entail? In my search for trying to find a generally agreed upon method for assay development, what I found is, not surprisingly, that there is no consensus for what validation requirements are or should be. However, in my 20+ years in assay development and my time running Ultivue's assay development and services lab, most seem to agree that at minimum, assay validation should involve accuracy and precision. In fact, I stumbled upon a paper summarizing a conference aimed at identifying best practices for biomarker development and validation, which was held by the American Association of Pharmaceutical Scientists in 2019. While there's no consensus on best practices for standardized validation requirements, of the 41 surveyed, 100% agreed that accuracy and precision assessment should be part of assay validation. However, accuracy and precision is only part of the story, as it oversimplifies the needed steps that are required to bridge from initial discovery to accuracy, and from accuracy to precision. And then once precision, marking the final step in validation, bridging to clinical validation. So for those reasons, I think validation is best encompassed in these 6 steps: optimization, accuracy, standardization, repeatability, reproducibility, and fit for purpose. So as I mentioned, the first step in validation is first the initial optimization. Optimization involves systematically adjusting and testing multiple conditions, and it's ultimately determined by intended use. In other words, optimization of an assay intended to be used for the identification of PD-L1 in vascular endothelial cells is going to have a very different optimization approach than an assay intended for identification of PD-L1 on antigen-presenting cells. Similarly, an assay intended to be used in conjunction with a threshold-based analysis approach is going to have a different optimization endpoint than one intended to be used in conjunction with AI-based image analysis approaches. In our case, optimization parameters are made even more challenging by the fact that we are optimizing to suit a wide breadth of use cases and a wide breadth of possible indications and image analysis approaches. So we do this first by careful antibody clone selection, comparing multiple clinically relevant clones, titering those antibodies, and identifying a condition that provides both highly specific target binding and the desired analytical sensitivity. And because our OmniVUE platform allows researchers to select any targets of interest to be combined into a ready-to-use kit, we must also identify the optimal fluor channels and concentrations to be used for each biomarker. Then we do this experimentally, first by testing each target in every single fluor channel in multiple fluor concentrations, and determining the optimal fluor concentrations for each target across multiple indications, both tumor and normal tissue. In other words, we do all the optimization so that we can provide an out-of-the-box solution that is immediately ready to be employed for research use. Now, while that particular consideration may be unique to our platform, a robust optimization approach really lays the groundwork for successful validation, and that's true irrespective of the assay. Next, we move on to accuracy, and that's accomplished by a predicate comparison to some known or accepted standard. In our case, that known standard is traditional IHC, which despite being invented 80 plus years ago, it's still the assay with the strongest foothold in clinical pathology. Pathologist reviews these assays qualitatively to ensure the correct subcellular location, the correct distribution, and of course, the correct staining pattern. So here we have an example of CD8 staining in human tonsil, where the expected subcellular location is cytoplasmic, and the expected distribution is scarcer in the germinal centers and more prevalent here in the interfollicular zone. You can also see that the IHC and InSituPlex staining here have a very concordant staining pattern. Now, once we have demonstrated that staining in InSituPlex technology is both accurate and optimal, we then must establish a set of standards to ensure that we keep that consistency over time. And we do this via qualitative and quantitative QC metrics. So each time a new lot of antibody conjugate is manufactured, that new lot of antibody conjugate is functionally tested against the previous lot. This ensures that qualitatively, of course, the staining pattern and subcellular location is correct. Then quantitatively, we ensure that the mean peak signal intensity meets at least 10 K, and that we have the desired sensitivity and specificity as quantitatively measured via the signal to background ratio, or SBR. That is, what we want to ensure is that our assay is sensitive enough to see a wide dynamic range of expression, but that it's, in other words, you're able to see very high expressors and very low expressors in the same area of the tissue, but that it's also specific enough that the non-specific or background staining is not accentuated. Here you can see a new lot of MHC2 antibody being tested against a previous lot, where the mean peak signal intensity for the new lot is 47 K, and the mean peak signal intensity for the previous lot is approximately 43 K. And that represents a 9% difference, where our accepted limit is up to 20% difference in signal intensity. Now we move on to one of the key components of precision testing, repeatability. Essentially asking the question of, can the same results be obtained under the same conditions? So same operator, same instrument, same lot of reagents, so very carefully controlled experiment. Here you see an example of this via single marker data sheets, demonstrating our serial sections of human tonsils stained as an intraday experiment, that is in one staining run. So this is cytokeratin SOX10 cocktail. And we calculated the coefficients of variance to be 6.3% for positive signal intensity and 2.9% for signal intensity. The other component of precision testing is reproducibility. So can the same results be reproduced, but this time under different conditions? This is particularly challenging when there is a highly configurable assay. And that's where we intend to focus the remainder of our time. What we wanted to demonstrate in these series of experiments is that one target, in this case, in this example, CD8, has equivalent performance in singleplex as it does in multiplex, and that it performs in the same way in different 4-plex configurations as it does when you compare 4-plex configurations to an 8-plex configurations. In other words, demonstrating that there is no steric hindrance and that the signal is not diminished when targets are co-expressed and co-localized. Finally, because our technology involves the gentle removal of one set of probes, the first 4 markers, and in order to exchange for the next set of probes used for the next 4 markers, we wanted to demonstrate that this fluor removal process didn't impact individual target performance. This further underscores the fact that really the validation approach must meet the operational need and the intended use of the assay being validated. Before we get into the results of the series of reproducibility experiments that we conducted, we should discuss the additional considerations for validation of a tissue-based assay. Namely, what tissue do you use and why? We used a 12-core microtissue array containing 4 types of tissue: tonsil, colon, lymph node, and melanoma. Each tissue type contained 3 cores from 3 distinct donors, and I'll go into why in just a moment. Something to consider when you are evaluating precision is that you're ultimately measuring not just the precision of that assay or the variability of that assay, but really you're measuring the cumulative variability from various sources. And that variability can arise from instrumentation. So the autostainer, you want to consider how the reagents are being dispensed. And if there's a known edge effect or gradient staining, you may notice that in our example, our configuration of the TMA, that the tissue types are interspersed instead of being in discrete rows or columns. And that's to safeguard against this possible gradient staining resulting from reagents not being dispensed uniformly across the entire surface of the slide. Another thing to consider is the scanner you're using. How are the images scanned and how can you mitigate out of focus images? It may seem like a good idea, for example, to to validate using a TMA that has 50+ cores, all with a very small diameter. But in doing so, you increase the likelihood of out of focus individual cores or discrete areas of the core. Other factors to consider, which I won't go into in any great detail, are tissue sample collection parameters. So variability, of course, can arise from the way that tissue is collected. Anything from cold ischemic time to how long in formalin to how embedding and microtomy were conducted, all can result in tissue loss or epitope masking or degradation. And then, of course, there's the inherent variability of biological variability of the tissue itself, some more than others. We all know, for example, that tumors tend to be more heterogeneous, have more heterogeneity than normal tissue. And you can see that's why we chose to use a blend of tumor and non-tumor indications. So the first phase of our reproducibility study was investigating singleplex to multiplex concordance. 10 multiplex and 8 singleplex slides were stained during the same staining run. Slides were interleaved so that single target performance of serial section 1 stained with a multiplex was compared to serial sections 2 stained with that same target in a monoplex format. The cell densities and signal intensity of each pair was then compared. Here, each dot represents a single core, and each tissue type is color-coded. the results show very high concordance of cell densities between singleplex and multiplex concordance, with each target's Pearson's and Lin's correlations falling at or above 0.9, where Pearson's correlation coefficients represents a linear correlation between the measurements, and a Lin's concordance correlation represents concordance of measurements. Similarly, when we move into intensities, we see the same trend. Here, each target's Pearson's and Lin's correlation coefficient once again falls at or above 0.9, with the exception of CD68, whose Lin's correlation falls at 0.8, which still represents a very, very high concordance. The next phase of our studies is aimed at demonstrating panel configurability. So we created 4 distinct multiplex panels, 2 where targets could be compared in the same staining channel in the presence of 2 other unique targets. In this case, panel A has CD8 and CD68, but then the addition of PDL1 and cytokeratin SOX10, where panel B has CD8 and CD68 with the addition of CD3 and Ki67. In the image above, you can see that CD8 and CD68 in all 3 panels, panel A, B, and D, qualitatively have the same signal distribution and cell densities, or approximated cell densities for those 2 targets, CD8 and CD68. The quantitative analysis confirms exactly what we were able to observe qualitatively, that not only is CD8 and CD68 highly concordant across panels, but that all targets demonstrate high concordance of signal irrespective of the panel configuration. Once again, all correlation coefficients falling at or above 0.9. So that allows us to have confidence that our assays can be configured without any detriment to the individual target performance. What's more is that we see that same trend when we interrogate signal intensity of the individual targets from one panel to the next. And here again, each target's correlation coefficients fall at or above 0.9. Next, we wanted to take a look at individual target performance when stained as a 4-plex versus performance when stained as an 8-plex. Additionally, because our technology utilizes that aforementioned 4 fluor channels that are spectrally distinct, that means that when you're staining more than 4 targets, it involves a fluorescent probe exchange process. As a result, we also wanted to investigate the target's performance of targets that are not subject to that fluor removal process known as exchange, and the same target after being subjected to that fluor removal process. In other words, what we call round 1 versus round 2 of staining. When comparing individual target performance from the 4 to 8-plex configurations, we again see highly concordant results, with each target's Lin's and Pearson's correlations falling at or near 0.9. You can also see an interesting trend here as you begin to note, as each tissue type is color-coded, there is a clustering effect of tissue type at and around certain cell densities per tissue type. And now we look at that same 4-plex to 8-plex configuration and look at correlations of intensities this time. And we again see a similar trend with correlations falling at above 0.9, with the exception of CD68 and FOXP3, which are at 0.8 and, 0.8, which still represents a very high degree of concordance. And now we move on to investigating the effects of exchange. So looking at CD8 when it was stained in round 1, not being subjected to any kind of exchange process, and CD8 stained in round 2 of an 8-plex when it was subjected to that fluor exchange process. And here again, we see all correlation coefficients fall at or above 0.9. Now, as we move on to investigating the effect of fluorescent exchange on intensity values, we see that all targets have, again, the same correlation value of 0.9 or above, save for FOXP3 and CD68, whose correlations fall at 0.8 and 0.7, respectively. I'll note that this still represents a very highly concordant signal. And by now you may have noticed that CD68 is often the outlier. This is not at all unusual. Since CD68 is a macrophage marker and is often, it's challenging to distinguish sometimes the dendritic cell protrusions that may be part of true signal from the non-specific background. It's also important to note that for all of the results represented here, that these are really raw data. That is to say that no tissues were excluded, irrespective of the validity in excluding them. In other words, even if they were 2 or more standard deviations from the mean, none of that, they were not excluded from analysis, nor were any exclusion annotations applied to any pores. As such, I would expect that with further refinement, we can expect improvement of these correlations beyond what is already very concordant signal. So now that I've shared the full breadth of our validation approach for OmniVUE, I wanted to go back to one of the key steps of validation for which I've not yet covered, and that's fit for purpose. So the term fit for purpose first appeared in a publication from the ligand binding analytical focus group in 2006. What it's come to mean is that assays should be validated for the intended use of the data and the associated regulatory requirements. It's not unlike optimization in that way, in that the approach and the scope of what's acceptable for validation is ultimately determined by the use case. An assay that is going to be used for clinical studies for exploratory endpoints is going to be vastly different than a validation approach for a clinical study involving patient stratification, and even different still than another one used for clinical diagnostic purposes. I wanted to demonstrate an example of one such clinical validation conducted in Ultivue Service Lab. So this particular researcher selected 8 targets to be combined into a custom 8-plex panel. And then once that panel was configured, they opted to conduct their panel validation with a focus on inter and intraday precision. And so here I have the schematic of the layout. In this case, 9 TMA slides were stained, each day representing a single automated staining run, where for the intraday repeatability or the interday precision, we were comparing pairs of cores from TMA 1 versus 4 and 4 versus 7, 1 versus 7, and 2 versus 4, and so on. Whereas for the interday repeatability, we were looking at pairwise comparison of cores across days. So in other words, comparing 1 versus 2 and 2 versus 3, and so on. What the results here show, this is an example of the plots for staining, where each core, again, is represented by a single dot. And it's an example of our CD4 reproducibility for this particular intraday study. And you can see a perfect 1.0 Pearson's and Lin's correlation coefficient. You break that down even further by target, intraday reproducibility for CD3, Pearson's correlation coefficient was found to be 1. For CD4, it was also 1. For CD68, 0.98. For CD8, 1. Cytokeratin SOX10, 0.98. FOXP3, 0.97. Granzyme B, 0.99. And PD1, 0.98. And then once again, for cell count reproducibility for an intraday study, we show similar results, so similar correlations between stains happening in distinct staining runs across multiple days, in this case, 3 days of experiments. So what does this flexible panel validation really empower researchers to do? What it allows is single markers tested in multiplex ISP assay showed expected localization and expression pattern. And it allows you, we demonstrated the lack of steric hindrance. We showed that a high level of reproducibility can be obtained irrespective of the other targets in the assay. How does this then help support your research? So really what it allows you to do is test multiple hypotheses and modify your hypotheses as needed. So you may begin by investigating TLSs, and then pivot to immune reactivity easily by simply removing CD8 from your panel, swapping in Ki67, and similarly removing CD20 from your panel and swapping in Granzyme B. And then your panel can be further trimmed down for higher throughput as you encounter larger cohorts. For example, you can take that 8-plex immune reactivity panel and whittle it down so that you're only focused on T cell activation. And you go from looking at 8 targets at a time to only looking at 4 targets at a time. Those 4 targets can then be whittled down further, or other targets can be added for additional insight. I wanted to thank you guys for joining me today for this talk. I will end here with gratitude for your time and take the opportunity to take any questions.
All right. Thank you so much, Yvette, for that great presentation. Now, now we will move on to answering some of your questions. Our first question is, if I choose my markers of interest and ask you to build my OmniVUE panel, what considerations will you need to take into account given everything you suggested for validation?
Yeah, I think first and foremost, it's intended use. At the risk of drilling that point home, it's you must consider what you're actually going to be using that panel for, and that'll help both determine how that panel is will be constructed, what targets are included, and ultimately define your clinical validation approach, or whether you need clinical validation at all. So we also have a scientific team here at Ultivue that's always willing to help to talk through panel design or validation considerations. Angela Vasaturo herself, who's helping field questions in the background, is the director of that team.
Right, great. Our next question is, how did you measure the intensity in these experiments specifically?
Yes, so it is mean signal intensity, mean signal intensity across the whole slide image. So I guess that's an important distinction to make, is that oftentimes what is utilized for image analysis is select regions of interest. This is the entire whole slide image with, as I mentioned, no exclusion annotations.
Okay, perfect. And can you comment on what the best controls are for MIF assay development, and what is your approach to account for tissue heterogeneity?
Sure. That is such a good question, and one which I can talk about with passion for many, many minutes, but I'll spare you. I am a board certified histotechnologist by trade, so I spent many years, really considering and refining approach to processing tissues and dealing with tissues. And there's really so much to consider with regard to tissue controls. But I think that the best approach is to use a normal tissue like tonsil, and that helps you gauge the optimal performance of the assay. And then ideally you include tissues that closely represent the tissues that you're going to be using later in the study. So that's easy to control with respect to indications. In other words, if you're going to be conducting a study that is based on non-small cell lung carcinoma, then of course you would want to include an NLCLC and a non-small cell lung carcinoma in your controls, in your staining controls. But it's not as easy to have a predictor of your staining performance if you're going to be receiving samples from multiple clinical sites, because those multiple clinical sites may have completely different processing methods, and those can contribute significantly to the quality of staining. In other words, they can, additional overly fixed tissue will have additional epitope masking and require additional epitope retrieval, or too little processing of the tissue can result in degradation of the tissue. So there's a lot to consider there, but ultimately at least a normal tissue and as close representative of the tissues you'll be using later on is best approaches.
Okay, that makes sense. Okay, our next question is, can you describe this is similar to the one I just asked, can you describe your QC methods for a given panel of choice and how you specifically control for a lot-to-lot variability?
Yeah, as I mentioned earlier in the talk, I talked about the individual conjugate QC process, but it's important to note that our fully assembled kits, so when an OmniVUE panel is constructed, they also undergo the same qualitative and quantitative QC process. So we stain 3 serial sections, and we assess the mean peak signal intensity and the signal to background ratio for that, for the average across those 3 slides. And then if there exists a previous lot of that particular panel configuration, then we stain an additional 3 with the previous lot. So 6 slides in total. If there's a previous lot, those slides are interleaved to help mitigate for any heterogeneity existing from the tissue. And the kits are measured for signal intensity, mean signal intensity, where no more than 20% variability is tolerated.
And okay, our next question. Typically, threshold-based image analysis tools are influenced by variable expression levels and tissue heterogeneity. How does your image analysis pipeline account for this variability in staining?
Yeah, so we utilize an AI-based, a deep learning-based approach. So unlike threshold-based analysis that is beholden to signal intensities as a means to define the cell positivity, this approach really utilizes all available features. So that includes cell morphology and cellular neighborhoods, And what that allows us to do is it really helps refine and mitigate any non-specific background or staining artifacts so that you get the truest representation of the signal. And then these algorithms or these models are trained on across multiple indications. So all of the tissues that are stained through our services lab. And that means that they're continuously refined as more and more slides are stained.
Okay, awesome. And do you think we should require standard guidelines for not only multiplex assay validation, but also for these image analysis pipelines?
We've had a hard enough approach coming up with standard approach definition for assay validation. But I do believe that successful validation, particularly for something that is so data heavy like multiplex immunofluorescence, really hinges on the methods that are used for image analysis, and not just image analysis, but the approach to data analysis. So I think it's tempting to make the pursuit all about making the numbers fall into a perfectly defined threshold. Instead, I would urge that we carefully consider what it is that what is it that you're trying to glean from the data, and what's the best approach to do that. So I guess to answer your question, I think that guidelines are helpful, but they shouldn't be so prescriptive that they stifle the ability to tailor the image analysis approach to be suitable for the intended use.
That makes sense. Okay, and we have another question. What is the timeline for shipping a kit, including all of this validation as you described it?
Yeah, I think that's the part. We've done so much of this work up front. As you can see, it's a pretty intensive validation process. And so what that allows us to do is build and ship a kit out, including all of this QC process, as I've described, both at the single level conjugate level and the kit level, and ship them from the date that the order is received to the date it is shipped out is 4 weeks or less.
All right. Okay, we have another question coming in. Are you able to combine OmniVUE staining with other types of staining like H&E that are commonly used?
Yeah, absolutely. That is one of the power of our technology. So our ISP technology is so gentle on the tissue that it does allow for same slide H&E staining. And that really helps because pathologists, as I'm painfully aware, are a creature of habit, right? Are creatures of habit, and so they rely on the H&E to really identify those structures where they can focus their attention, and it really also helps define. It also helps confirm like tumor stroma stratification. So you're not just simply beholden on a biomarker like cytokeratin to help identify tumor versus stroma areas.
Okay, great. Okay, let's see. We have another question. What kinds of customers or kinds of research are you guys usually receiving when you get, for example, an assay services project? What kinds of research does that involve?
Yeah, so typically we are in the we stem anywhere from early or late phase exploratory research, and what I'll define that as is there's that very early discovery phase where you're throwing all the biomarkers to the wall in hopes that something sticks. And then as you whittle down your hypothesis, you're moving into a mediumplex or translational research mediumplex phase. And then we also support preclinical research. So we have both mouse-specific panels as well as NHP-specific panels that can be employed for preclinical studies. And then as you're moving into that translational space where you're moving out of that, casting the wide net discovery space, and you whittle down your hypothesis, we tend to be in that early phase clinical studies where where it's more about exploratory endpoints.
Okay, gotcha. And how can you go into a little more depth about how those 24 biomarkers were chosen? What were they most common, most prevalent, most important?
Yeah, so these, that's a great question. So these are biomarkers that are pretty ubiquitous in immuno-oncology, and that's kind of where we began our focus. Although what we're finding now is that these biomarkers are really relevant and important to multiple different areas of focus beyond just immuno-oncology. And so, where there's inflammation, there will be these biomarkers where and that applies to many different disease states and areas of focus for many different types of research, including autoimmune disease and neuro, and we're finding more and more indications or more and more areas of research that can be aided by those 24 markers.
Okay, and if I had a panel that was totally out there, not really well-known biomarkers I wanted to look into, would Ultivue be able to help me with that work as well?
That is a great question. One to highlight here for sure that we are the 24 targets that we had that are well validated are not the only targets that we've had experience with, and not the only targets that we will have experience with in the future. So we have to date we have to date optimized. So we have at least had experience with approximately 175 targets. And 300 plus distinct clones. So for example, we have our well-validated PDL1 clone, 7310. If a customer has their specific PDL1 clone that they've already begun some research with or on, or they maybe are moving towards a clinical path and they want to make sure that it's the CDX clone, we can, for example, incorporate that into their research and optimize and validate as necessary in support of their studies. So any target of interest, if it exists, if you've made it, if it exists in the commercial realm, we can optimize it and make it work in a panel.
Okay, and you mentioned that the OmniVUE kits will, door to door will be about 4 weeks. What is the timeline for this more custom biomarker panel?
Yeah, we've definitely refined our approach to optimization, and we've given a lot of thought in making improvements both to the breadth of the experiments and making sure that they're thoughtful in order to get the best specificity and sensitivity in the shortest amount of time possible. So also because we are direct immunofluorescence, it really simplifies the amount of levers that you need to pull. And so I'm very happy to say that custom development is usually completed between 4 to 6 weeks. Okay. So each individual marker can be optimized between 4 to 6 weeks.
Okay, for each individual one, and then combined, it'll be however long that is.
Correct. Sometimes, depending on the number of markers they are, much of that work can happen in parallel. So if you have an 8-plex panel with 4 unique targets, your custom targets, that work can happen in parallel. So it's not an additive effect. And then the panel development usually happens very quickly thereafter.
And that includes image analysis and making those optimizations as well.
So the image analysis is a separate endeavor. Image analysis, for anyone who has ever conducted image analysis, is quite an undertaking. So image analysis timelines are always going to be longer than assay development timelines, or about the same, I should say. That being said, we are making such great strides in defining and refining these image analysis algorithms that over time we will be higher and higher throughput as these models are better and better trained.
Mm-hmm. Okay, and you mentioned in a previous answer that in addition to human panels, Ultivue also offers mouse panels and non-human primate panels as well. Can you go into more depth about what that looks like for Ultivue?
Yeah, so I think we don't validate those targets in the same way that we have described here. That being said, we have I believe about 15 mouse targets that we've had experience with, and always willing to consider developing more. And similarly with NHP targets, we have a larger range of targets, individual targets that you can select a list from that have already had experience with, or come to us with any target that may not have had experience with, and we're willing to do custom assay and panel development for those.
That's great. And as far as assay development goes, can I actually do one part, like the staining, for example, and then take my stains and give them to Ultivue for image analysis, or is it a complete pipeline? How does that work? Can you segment your services?
Yes, so that's really important point to make too, that we do have three distinct really arms of our company. So there's two distinct services arms. There's the assay development and the staining, and then the image analysis. So even if you have done your own staining, let's say you purchased a kit from us or done some multiplex staining elsewhere, you can submit your images to us via a platform, and we can provide image analysis support.
Okay, great. Well, I think we're coming to a close. I want to thank you again, Yvette, for your amazing presentation and an awesome Q&A session. If we didn't get to your question, or if anyone thinks of a question that they would like to ask, please visit ultivue.com or reach out directly to Angela at angela.vasaturo@ultivue.com. Stay tuned for more webinars to come and subscribe to our email list to stay up to date. And thanks again to Yvette and Angela, who has been helping with questions in the background. And thank you all for attending today's webinar. Take care.